DNA structure. DNA is a long chain in which is located on every cell of our body. It is formed of nucleotides. The DNA information is stored from four nitrogenous bases: adenine, guanine, cytosine, and thymine. In every nucleotide contains a phosphate, a sugar, and nitrogen base. In every sugar, there is 5 carbons named deoxyribols. And actually, the order in which the nucleotides are organized is important, just for the reason that the order depends on genetic code. Cytosine and guanine and adenine and thymine are bonded with hydrogen bonds. Adenine and thymine have double bonds, and cytosine and guanine are triple bonds. The structure of the DNA is a double helix, such as a twisted ladder. This set of DNA structure have the same amount of nucleotides that the RNA. The difference between the RNA and DNA is that DNA is a double helix and RNA is a single helix. The DNA contains the instructions that our organism needs to continue working as normally it does.
DNA replication. DNA replication is a very important process, and it happens every 8 hours in your body. It all starts in the cell cycle. To be precise, in the interphase, not in the mitosis. It happens inside the S phase of the interphase. The first thing we need to know is that we will split the DNA with the help of the helicase. Also, the proteins will take care of not letting the DNA get together again. The two strands will be leading and lagging. Then, we will have to name the primes. We have from 5 to 3 and from 3 to 5. This consists on the position of the sugars. Counting clockwise, we have the 1, 2, 3, 4, and 5 outside. And if it's upside down, 1, 2, 3, 4, and 5. Now, the primase produces a primer to start synthesizing the DNA. Now we have the polymers, which walks by adding the missing and exact bases to the DNA strand. But here it gets a little tricky, because the polymers can only walk in one direction and entirely complete only one strand. That goes from 5 to 3, which is the leading. We have the lagging, which goes from 3 to 5. This means that the polymers, as long as it walks on by, it will leave some chunks along its way, which will be called Okazaki fragments. After all this process, it comes the ligase, which finally bonds all the bases with its due pair, adenine with thymine, which are double bonds, and cytosine with guanine, which form triple bonds. And that's it. And now we have 2 new brand strands of DNA.